Hey guys, welcome to your lecture on sensation, and this one specifically is going to be about vision. Vision is going to be our dominant sense, the thing that we rely on the most and the thing that we choose to utilize the most. When we talk about the visual system, we're going to be looking at what is called the visible light spectrum. And for humans, that's anything that's a little less than 400 nanometers to about 700 nanometers. When you hear me say the word nanometer, I am talking about a unit of length, so billionths of a meter, super, super short in length. Length is going to become important. When we talk about how we perceive color, the actual hue, by the length of the wave. So here, take a rainbow and flatten it out. If you were to do that, purple would be much shorter than red. This is nature really showing us the length of waves. Red wavelengths are somewhere in the 700 nanometer range. They're much longer than maybe our purples and our blues, which are somewhere closer in that 400 range. The other thing that we know is that the taller the wave, and again, we were talking about a light wave, the taller the wave, the brighter the color. It doesn't change the hue of the color, it just makes it brighter. So you can have a bright red or a dull red. If it's a dull red, the wave is shorter. We're going to talk about how a light wave becomes a neural message. So you guys should hopefully understand that when we were talking about the brain and the neurons, neurotransmitters, and neural firing, that's the only way that the brain understands messages. Hormones and neurotransmitters, nothing else. So when we're talking about light waves are not processed by the brain. That means that our eyes don't really see. Our eyes are just there to convert. The first structure that we're looking at in this conversion is the cornea, the clear front part of your eye. If you wear contacts, this is where you're putting your contact. It's curved to help better focus and bend light waves so they make it to the back of the eye, and it's protective. It's also highly sensitive. If you've ever scratched your cornea, you know what I'm talking about, but it's the area that first is struck by light. Next, we have the iris and the pupil, and they go hand in hand. The pupil is a hole, and the iris is what makes that hole large or small. The iris is just a ring of muscles, so if you tell everyone you have brown eyes, it's because you have a brown iris. If you have blue eyes, it's because you have a blue iris. Now, blue eyes are a mutation. They are a deviation from what the norm is, which is brown eyes. And people with blue eyes are actually more light sensitive. The blue of their eye doesn't filter out as much light. Brown eyes tend to do a better job of protecting the retina. Your lens is going to be right behind your pupil and your iris. And essentially, it's this nice flexible structure that's held in place by muscle. It goes thin or fat to help focus light waves. Up until this point, everything has been about focusing light waves to get to the very back of the eye in a structure called the retina. Now, the retina lines the back of the eye, and the important thing about the retina is that this is the structure that holds the cells that turn a light wave into a neural impulse, that change the light wave into something your brain can read. When we're talking about our vision and focus, usually we only think about the lens, but it's the lens and the retina working together. If you have normal vision, your lens focuses an image right on the sweet spot of your retina, on an area called the fovea, and you see clearly. Now, if you're nearsighted, the problem is that your lens doesn't actually push the image far enough back to reach the retina, so it stays blurred. If you've ever used a camera and you are allowed to actually dial the focus, if you go out too far, everything's blurred, and if you come in too close, everything's blurred. There's just a right spot. Well, with people who are nearsighted, the only way that they can get that candle image on the back of the retina is to pull the image closer, hence nearsighted. Well, the opposite is equally true for farsighted. Farsighted, you notice that the candle is falling behind the retina. Well, the only way to make it fall right on that sweet spot is to pull it further away. And this is your retina. Now, hopefully you're seeing at the lower left-hand part of your screen that that's the eyeball. And you're seeing the light wave traveling through the cornea, the pupil, and the iris through the lens, and it's going to the very, very back of the eye. Your retina has 3 layers. The light wave travels all the way to the very first layer at the very back of your eye. Now, this is going to make sense here in a little bit. So, the very back of your eye is going to be your photoreceptor cells, and you see this up here labeled as number 1. Then the light wave travels to layer 2, so it's coming back to the interior of the eye. And those are what we'll call your bipolar cells. Then it travels to layer 3, which is the most interior layer of the retina in the eye, and those are your ganglion cells. your ganglion cells will form your optic nerve, and then you're going to see your optic nerve come all the way back out through the eye. That means that the optic nerve has to cut through layers 3, 2, and 1 to leave. And that's going to be really, really important here in a moment. So the receptor cells that we were talking about, those photoreceptor cells, and again, here we're looking at a cross-section of your retina in the picture. Your rods and your cones are your photoreceptor cells. They're at the very, very back of your eye. Then you have your bipolar cells right in the middle and the ganglion cells closer to the interior of the eye. Photoreceptor cells do what we call phototransduction. They take that light wave and they turn it into a neural message. Rods are going to be the photoreceptor cells that you have to the periphery of your vision and interspersed in the back of your vision. And they help you with black and white and gray, so those kind of different scales. And then it also helps you with motion. And it's really good in low light conditions. In other words, it's active when there's not a good strong light source. Next, we have our cones, and our cones are still in that photoreceptor layer. They're pretty much concentrated right in the center of your eye, where you best see. And so they deal with color, really kind of clear and sharp detail, and they need a lot of light. So if you don't have a lot of light, your vision gets a little fuzzy. If you don't have a lot of light, you don't perceive color very well. Light is very important falling on our cone to activate those cells. So they need a strong light source to work. When we look at something and you look at, say, the back of your hand and you can see the veins and maybe even the kind of pattern of your skin cells and the hairs, and that's falling right on your fovea and specifically on your cones. That's where you're getting all that detail from. And if you notice as you're staring at the back of your hand that maybe the desk behind it or the images around it are blurry or maybe slightly fuzzy, it's because they're not falling on that fovea. They're not being hit by mostly cones, and they're being processed somewhat by the rods that are in your eye. So they're giving you kind of a general sense, but no real clear detail. So what's the difference between rods and cones besides the fact that one sees color and detail and the other one does essentially light and dark and movement? Well, the number is also important. We have far more rods than we do have cones. We have 6 million in each retina for cones, and way, way more. We have about 120 million rods in each retina. Your rods are going to help you detect things like motion out of the corner of your eye. And the question is, well, okay, if rods do black and white and we don't have really cones at our peripheral vision, why is it that I can see color in my peripheral vision? And the answer is, your brain is lying to you. Your brain knows what to anticipate for the color in your peripheral vision, so it essentially makes you hallucinate it. You have expectation, and later when we get into perception, you'll understand what we're talking about. The bipolar layers are that middle layer, and they essentially do a refining process for the neural message. So your rods and your cones get stimulated by a light wave, and they turn the light wave into a neural message. They send that neural message down the axon, and that message is then received by the dendrite end of the bipolar cells. The bipolar cells then deal with the message and send it down their axons, and it's collected by the dendrite end of the ganglion cells. The ganglion cells send that message down their axons, and this is where it's really important. The axons of the ganglion cells, that third layer, that most inner layer, they form your optic nerve. Your optic nerve is the nerve that sends that neural impulse, that entire message about what you've seen. It sends it out of the back of the eye to the brain. So here we go again. The optic nerve carries that information from the eye to essentially the thalamus first, because it doesn't go straight to the occipital lobe, and from the thalamus then to the occipital lobe of the brain, the area of the brain that processes vision. But in each of your eyes, you have something known as a blind spot. If that nerve has to travel through 3 layers of tissue, there can't be rods or cones at a certain spot in your vision to process color or detail. And if those cells aren't there, there absolutely is no vision. So hopefully we'll get it to work in class where I can show you where your blind spots are. But your blind spots literally are in each eye. Now, because you see with 2 eyes, we have binocular vision. My left eye makes up for the lack of vision in my right eye, and my right eye makes up for the lack of vision in my left eye. So we're constructed in a way that we make up for this small little spot that we don't see in our vision. We've been talking about color vision, and we talked about cones. Cones detect color. Well, there are 3 specific cones that we have. We have a red, a blue, and a green. And this creates what's called the trichromatic theory. the three color theory, trichroma, three color. So we have large or long cones that detect red, we have medium wavelength cones that detect green, and we have short wavelength cones that detect blue. Notice there are no purple or teal or yellow cones. There's not even orange cones. We literally have long, medium, and short wavelength, and they are known as the red, the green, and the blue. We will talk about primary colors. Now, in art class, you guys talk about red, blue, and yellow as your primary colors. If you were to add red, blue, and yellow paint together, red, blue, and yellow crayons together, red, blue, and yellow colored pencils or markers, and you were to overlay them, you would start to block out the actual wavelengths that are reflecting off the surface. And therefore, you would have this kind of total absorption, and we call that the subtractive process. When paint pigments absorb the different lights, or wavelengths, sorry, that's subtractive. So if we're talking in paint or markers or pigments, it's subtractive and you'll end up with the color black. However, when we're talking about light waves, we're talking about a blue light, maybe with a red filter, and then another one with a green filter. If we overlap a blue light with a red light, and then overlap it with a green light, what you should see in that picture there, when we add them together, you get white light. But if you were to add maybe a green spotlight to a red spotlight, you should get yellow light. And so now we're starting to see, oh, red and blue simultaneously stimulated gives us our spectrum for purple. And you could essentially mix all of these colors in some varying degree to get every color that we see. That's the additive process. By adding light wavelengths to each other, if we add the big ones, red, green, and blue, we'll end up with white light. So how do we know that these three cones exist? Well, one of the ways that we know is because we have people who are colorblind. Usually people who are colorblind are what we'll call dichromats, meaning they have two cones or two colors. They often have difficulty distinguishing between colors. They're not truly blind to all color. They're usually lacking one of the cones, or one of their cones is overlapped and doesn't get a good processing of color. Most people who are colorblind very easily adapt to their environment, and they don't really notice their color blindness. They may have people correct them, they may have colors that they don't necessarily distinguish too well, but in the most part, they do fine. Most people who are colorblind are male, and that's because it's an X-linked trait. In other words, if you have it on your X chromosome, you're probably going to express it. If you're female, you have two X chromosomes, and for females, you actually have to have it on both chromosomes to have it. This is called the Ishihara test. This test is what is commonly used by ophthalmologists and optometrists to determine whether or not someone's colorblind. Unfortunately, in my class, I've had at least two students figure out that they were colorblind because they couldn't see the 12 in the circle or the 26 in the circle that's off to your right. One of those students, who I love dearly and is just an amazing person, made it all the way to her senior year without ever knowing that she was colorblind. We got her a pair of color correction glasses, and for the first time in her life, she understood what people meant when they said things like the leaves are changing colors. She told me she always thought it was just a weird thing people said in the fall. Well, here's why. Down in that lower picture, you see somebody who would see off to the left the leaves changing color in the fall. And you get that explosion of reds and oranges. Well, unfortunately for her, she saw what you see off to the right. The leaves didn't really change color. She was actually going to be the third row down in the different types of color blindness. So if you see normal and then protanopia, she was deuteranopia. And so for her, she was lacking in kind of the red-green spectrum. So everything was kind of good for blues, but not so great for the reds and the greens. So if you were looking at the apples, you would see a green apple, a weirdly fleshy colored yellowy apple, I don't know what color that is, and then a red apple. And if you look at the bottom, her green and red don't really differentiate much. She made it through her senior year because we write the names, the colors on crayons and pencils. And if you were driving a car, your traffic lights are always in the exact same order. So while people can be colorblind, it's not always the most limiting thing, and you can make it pretty far in life sometimes without ever knowing it. So we have the trichromatic theory, the three cones. But here's the thing. If we have red, green, and blue, how do we see yellow? And why don't we see a color called reddish green? Well, there's a reason for that. There's another theory that goes with trichromatic theory called opponent processing theory. And this theory states that when you stimulate one color, you're probably suppressing, or you actually are suppressing, another. If you stimulate your long photoreceptors for red, you have to then suppress your green photoreceptors. They can't fire. And if you're firing for green, you're suppressing for red. So you cannot see red and green blended together. The same thing with blue and yellow. Now, I will tell people we don't have a color named bluish yellow or yellowish blue, and they'll go, yeah, we do, it's green. No, we have a color called green. And we have a yellowish green and a bluish green, but we don't have a bluish yellow or a yellowish blue. And that's because if red and green are being simultaneously excited, we see yellow, and that suppresses our short cone for blue. But if the short cone for blue is being excited, then we suppress red and green. In other words, you can't see blue and yellow blended together. And then it's the same thing for black and white. In class, we'll play with some after images, but if you've ever sat in class and stared at a PowerPoint presentation for a long time, and then looked away and noticed that it felt like there was a big black square following you in your vision around the room, you're seeing what's called an after image. You stared at something that was bright white for long enough that it fatigued the photoreceptor cells in your eye. And when they got fatigued, they had to take a break, that refractory period that all cells go through, and their opponents, which would be the ones that would see black, were allowed to fire. We often don't have after images because we move our eyes a lot and we continue to refocus. But if we stare at anything for too long, we should see the after image. And that leads us to our last point for this video. Our eyes are always moving. So if we can hold them still, or if we focus too long on something, we can see some pretty cool explanations for how our eyes work. If our eyes were to stop moving, sense receptors would fatigue, images would actually vanish because our eyes would have to go through that refractory period. The cells would have to reset. So we constantly move our eyes to refocus and to re-see what it is we're looking at. Even when we feel like we're staring at something, your eye is just twitching back and forth at the smallest degree. We'll look at some illusions in class that hopefully should kind of show you how our eye movements impact what we see.